Students, today we are going to discuss about the next mechanism of the aromatic nucleophilic substitution reaction, and this mechanism is ARSN1, that is called aromatic nucleophilic substitution unimolecular mechanism. So basically, first of all, this mechanism is a very rare case, and generally it is observed in case of benzene diazonium salt. On the screen you can see some reactions in which the N2 is eliminated out from benzene diazonium salt, and this is being converted into different type of the products. So these reactions are aromatic nucleophilic substitution reaction, and these all reaction will proceed with ARSN1 mechanism. In ARSN1 mechanism, in the first step, which is the slow step, rate determining step, first of all gaseous nitrogen will be eliminated out. So this is the first step in which the gaseous nitrogen is being eliminated out from H1, and this is being converted into the cation, right? Phenyl cation will be synthesized. After formation of this phenyl cation, it will be attacked by the nucleophile. So here your nucleophile is a case of Y, and where Y can be chlorine, bromine, it can be N3, right, and so on. So it will be attacked by the nucleophile, and after attack of the nucleophile, it will get converted into the desired product, right? So this is the case of aromatic nucleophilic substitution, the unimolecular, right? When we talk about the evidence in support of the mechanism, the best evidence in the support of this reaction is the rate of reaction. That is, the rate is dependent on the substrate, that is benzene diazonium salt, which tell you that the reaction is particularly unimolecular in fashion. So this is the ARSN1 mechanism, and the driving force for this reaction is I have already told you that the elimination of molecular nitrogen, gaseous nitrogen, is the driving force for this kind of reaction. This is a case of ipso substitution reaction in which the nucleophile will be attacked at the position which position is already occupied by the leaving group, right? So this is a case of ipso substitution as well. So this is all about your ARSN1 mechanism, right? Now the third mechanism, yesterday we discussed about the ARSN2. Today in the first slide we discussed about ARSN1 mechanism, and now we are going to discuss about the benzyne mechanism, the third mechanism of aromatic nucleophilic substitution reaction, that is benzyne, which is called as the elimination addition mechanism, in which first step is the elimination step, and that step will be followed by the addition. So ultimately you will get the substituted product in which one nucleophile will substitute the another nucleophile on the aromatic system, that is aromatic nucleophilic substitution reaction. For example, on the screen you can see two reactions in which we are having a chlorobenzene. I have already told you in my previous lecture that the aromatic halide, right, aryl halides are generally very less reactive, or you can say that they are inactive for the nucleophilic substitution reaction. Reason being that this particular carbon and halogen bond is very strong and it cannot be broken down very easily. But if we use very harassing condition, if we are using very drastic conditions, right, for example we are using high temperature, we are using concentrated base, at that time it can be substituted by different type of the nucleophile. For example, this is the case in which we are having a chlorobenzene, and under very drastic condition at 350 degrees Celsius temperature and concentrated sodium hydroxide base followed by the hydrolysis, this particular chlorobenzene is getting converted into the phenol. So this is the nucleophilic substitution reaction. And how this kind of mechanism, if we are particularly talking about the benzyne mechanism, that is the elimination addition mechanism, for example this is the case, right, in which we have used soda amide in presence of liquid ammonia followed by the hydrolysis. In this case, the mechanism will be the elimination addition mechanism. we can see that two different kind of products will be synthesized in this case. So formation of these two products is mainly because of the intermediate that have been synthesized in this mechanism. When we talk about this mechanism, we should use the isotopic labeling experiment for elucidation of this type of mechanism. What we did, basically, the elimination addition mechanism will be the first step. Elimination means in which this particular group will be eliminated and there is formation of the intermediate, and the second step is the attack of the nucleophile on the position vacated by the leaving group or at its adjacent position, because we are getting this product. This is the ipso substitution, means the place on which the leaving group was present. We are also getting this particular product, which is the position adjacent to the leaving group, right? So basically it is proceeding with an intermediate, and that intermediate is called as the benzyne intermediate, benzyne or arine simply called as. And how we came to know about this formation of benzyne, or if we are particularly talking about the evidences about this mechanism, so the first evidence that prove that this particular reaction is proceeding with the formation of the benzyne is the isotopic labeling experiment, in which this is the carbon which is isotopically labeled, and when this chlorobenzene is subjected to react with soda amide in presence of liquid ammonia, there are 50% chance of formation of this product, means the product in which the nucleophile is occupying the position, is occupying that carbon which is isotopically labeled, and the 50% chance is that the particular nucleophile is occupying the adjacent position to the isotopically labeled carbon. How we can see it? We can see that in the step, first of all, this particular NH2 negative will abstract the proton, and this will form C negative, which further will form the benzyne with the elimination of this particular chlorine. So in this benzyne, we can clearly see that this is the isotopically labeled carbon, and this carbon is not isotopically labeled. So when the nucleophilic attack will occur, the nucleophile can either attack at this position as well as that can attack at this position. So there are 50% chances that it can attack at this position as well as it can attack at this position. So this is the first evidence regarding the benzyne mechanism, regarding the formation of the intermediate arine or the benzyne in the course of mechanism, right? If we talk about the second point, means this is your first step of the mechanism in which benzyne is being synthesized, and we talk about the second point that it is attacking here. So this will be the carbanion which will abstract the H and will get converted into this product, and this is the chance in which it is converted into that kind of product. So this chance is with equal probability. Now we come to the second evidences about the mechanism, that this mechanism is the benzyne one. See, this is the substrate in which this is your bromine, this is your leaving group, and these three are the substituent attached to this particular substrate. We can see in the mechanism that this particular hydrogen, that is the hydrogen attached at the ortho position. That is, this is your ortho position. So this carbon is bearing the hydrogen. So this H positive will be eliminated out. Means the necessary condition for this mechanism to happen is there should be a hydrogen at the carbon ortho to the leaving group. So in this case, as there is no hydrogen, so when this particular substrate is subjected to react with KNH2 in presence of liquid ammonia, there will be no reaction at all, because this particular substrate cannot form the benzyne intermediate, that cannot form the arine intermediate. Reason being, the first step will not be possible in that case, right? So there is a requirement of this particular hydrogen. So it is a strong evidence that will prove that the mechanism for this reaction is through the formation of the intermediate benzyne. Now, if we observe the case of this particular substrate, for example, this is a substrate in which this is your leaving group and this is the ortho position, because this ortho position is not having any hydrogen. So this KNH2 first of all will abstract the hydrogen from this position, and this will form benzyne at this particular between these two carbon, right? So the second step will proceed with the attack of NH2 negative either at this position or at this position. Means we are getting the ortho product as well as we are getting the meta product, but there is no para product in this kind of substrate. So known formation of this product during the course of reaction is a strong evidence which indicate that the mechanism of the reaction is through the formation of the intermediate benzyne, because you know that this is the ortho position and hydrogen will be eliminated from here. So the benzyne will be formed between these two carbon and there is no possibility of formation of benzyne here. And when the benzyne will be formed here, at that time the para product will be synthesized. But here you are getting the ortho and the meta product only, because you have only the possibility of this benzyne, right? So this is a strong evidence regarding this one. Now if we talk about the benzyne, when we see it in first instance, we can see that it is containing carbon-carbon triple bond, so it will have an alkyne-like character. But when we observe the molecular orbital diagram of this particular benzyne, we came to know about it that this is a very unstable type of intermediate that have been synthesized during the course of reaction, and because of side-by-side overlapping, right, it has a triple bond like of character in which we are having side-by-side overlapping, and this is generally, you know, appear as a triple bonded form. So this is the normal pi bond and this is abnormal pi bond between this one, that this is the formation of this one. And we can see that two sp2 hybridized orbital which are outside the ring, they will overlap with one another and will give rise to this unstable type of intermediate, which is called as the benzyne. And when we talk about this benzyne mechanism, and when we talk about the strong evidences regarding the mechanism, that whether this benzyne has been synthesized during the course of reaction or not, we came to the conclusion that when if we talk about in your previous semester you studied about the reaction intermediates, and the benzyne you have already studied about it, that it will undergo dial center type of reaction. Means when this kind of reaction will proceed, at that time when furan is being added into the reaction mixture, and we know that the mechanism is proceeding with the formation of the benzyne, so there will be a Diels-Alder reaction between the benzyne and the furan, and we could observe this cycloadduct between the reaction between this furan and reaction between this benzyne, which is the Diels-Alder reaction. So this is again a very strong evidence which prove that this kind of reaction, that is the elimination addition mechanism, will proceed with the formation of the benzyne or arine intermediate, right? When we talk about the benzyne, the first major evidence for the presence of this particular benzene intermediate, it was given by J.D. Roberts in 1953, right? And it was, with the isotopic labeling experiment in the bromobenzene. So this particular benzene was discovered with this particular experiment, right? Now, what will be the stereochemistry of the product? What will be the direction, if we are particularly talking about the direction, or if we particularly talking about the major and the minor product formation in this mechanism, then how we can predict that which product will be synthesized during the benzene mechanism, aromatic nucleophilic substitution benzene mechanism. So when we talk about this first category, for example, category number 1 is, there is electron withdrawing group like NO2, like CHO group, carboxylic acid group, like sulfonic acid group is here, right? Like CN group is there. So if we are having electron withdrawing group attached to the substrate, and in that case, this is the arine, this is the benzene that have been formed, means between para and metacarbon. And when the nucleophilic attack is allowed on this particular benzene, this nucleophile can attack at this position, and this nucleophile can also attack at this position. So which will be the major product and which will be the minor product, we can identify. How we can identify? When the nucleophile is allowed to occur at this para position, at that time, this is the carbon that will bear the negative charge. When this nucleophile is allowed to attack at this position, this is the carbon which will bear the negative charge. Now, when we talk about the stability of the carbon ion that have been prepared, we can see that this is the electron withdrawing group, and this is the carbon bearing the negative charge, and here this is the electron withdrawing group, and this is the carbon bearing the negative charge. When we talk about the distance, we talk about the stability of this particular carbon ion, we can see that this is very much stabilized as comparison to this one. So this will be the favored attack as comparison to this one. So it will be the major product when we talk about the first condition. Case number 2, when we are having electron withdrawing group and the benzene is allowed to be formed between this orthocarbon and between this metacarbon. At that time, we can see that either this nucleophile can attack at the metacarbon or this can attack at the orthocarbon. When it will attack at the metacarbon, it will be the carbon ion, and when it will attack at the ortho position, it will be the carbon ion. So when we talk about the stability of this particular carbon ion, this is the stabilized position as comparison to this one. So again, this will be the favored position of attack as comparison to this one. So this will be the major product in that case, right? So when we talk about this one, we can clearly identify that where the nucleophile will be positioned, where the nucleophile will attack. That is the direction of formation of benzene and direction of formation of the product, right? Now we can see that this is your para-chlorotoluene, and when para-chlorotoluene is subjected to react with soda amide in presence of liquid ammonia, at that time we can see that this will be the benzene. This will be the benzene that have been prepared. Now we know that CS3 group is electron donating group, right? So you can have this para attack and you can have this meta attack, right? When we talk about this particular para and meta attack, you know that at meta attack the C negative will be here, and when we talk about this particular para attack, the C negative will be here, right? So you are getting these two type of the products in which one will be in the major quantity and the second one will be in the minor quantity. You will observe in that case, right? Now when we talk about CF3, for example, this is your another substrate CF3, and CF3, this is electron withdrawing group, and again you are getting this kind of benzene. So when you talk about the ratio of these two products that have been synthesized, as I have already told you that this is the preferred site of attack as comparison to this one. So the major product will be this one as comparison to this one. So the ratio is about 60 to 40. When OCS3 group is present at the para position to the leaving group, again you are getting this particular benzyne, and in that case you are also getting the para product as comparison to the meta attack, right? So this is the electron withdrawing group, and because of this region it is the direction of formation of benzyne, and according to that direction the product will be synthesized. Now we are having CS3 group which is attached with this one. This is your orthochlorotoluene. When we talk about orthochlorotoluene, that this is the benzyne that have been synthesized, right? And when we talk about the product, now this can attack at this position and this can attack at this position as well. So when it will attack at this position, it will be the C negative, right? And when it will attack at this position, it will be the C negative. So when we talk about the major product, however, there is no strong preference about the CS3 group when attached to the benzene ring. But when we generally talk about the major product that have been synthesized during this reaction, that will always be that this will be the major product as comparison to this one. For example, this is your CF3 and this is your chlorine. So this will be the benzyne, that this will be the only benzyne that have been synthesized, right? So when we talk about this one, it can attack at meta position as well as it can attack at ortho position. So the meta attack will be favored attack. When we talk about this one, again this will be the benzyne that have been synthesized, right? So again the meta attack will be the favored attack in that position. So when we talk about the direction of formation of benzyne, and as well as when we talk about the formation of the major product, we can see that what kind of benzyne can be synthesized, and from that benzyne what kind of product can be synthesized. So that will depend upon the group which is already attached on the benzene ring. So electron donating group and electron withdrawing group, they will how they will stabilize the C negative that have been synthesized during the second step, right? So that will determine the formation of the major and the minor product. So this is all about your aromatic nucleophilic substitution reaction that is proceeding through Benzyne mechanism and the aromatic nucleophilic substitution unimolecular mechanism. Tomorrow we will discuss the last mechanism and some named reactions of this chapter.